Hello, everyone. I hope you're doing well. We will finish up lecture 7 today on special senses. It's a pretty short lesson. Probably will not have to go all the way to 11 o'clock. But you never know. I always say that and then I end up going way over time. In any case, let's get started. So last class, we labeled the eyeball, and this is another diagram that that you can look at. Like I said last time, right? It's a good idea to just search up various diagrams online and see if you can identify the same thing. And personally, I found that very helpful in terms of helping me. A, recognize the parts, and B, just seeing it from different perspective really helps consolidate the learning. Okay, so one of the first thing you should remember about the eyeball is that there are 3 layers. Okay, so we have the outermost layer, which is the sclera. Beneath that is the choroid, and then beneath that is the retina. So now we're going to take a closer look at these 3 things. Starting with the sclera. The sclera is the most superficial layer of the eye. It's the outermost layer, and it's the white part of the eye. Okay, when you look at somebody's eye, you will see the color part, and then surrounding it, you will see the white part. So that's the sclera you're looking at. And in this very zoomed in eye right here, where my mouse is, that's the sclera. It's white and it's fibrous, right? It's created like a kind of like a tough a casing for the eye, right? Now, I don't know if you ever get poked in the eye either by some tree branch accidentally when you're walking on the sidewalk, or like you just accidentally poke yourself in the eyeball, but you're not gonna make a dent in the eye, right? So the sclera is quite tough, quite fibrous. Okay, and it provides a place for eye muscles to attach to, right? So you actually have a lot of eye muscles connected to the eye, and it allows our eyes to move virtually in any direction. So let's say this top one, right? You don't have to memorize the muscle's name, okay, obviously. So we have the, by the way, this eye, anybody want to tell me which eye this is at just by looking at the name of the muscle? Is this the right eye or the left eye? And how do you tell?
Right?
How do you know that, Leah?
So medial is close to the midline of the body, and lateral is outside.
Yep, you got it, right? Very good. So that's medial right here. That means your nose is going to be here. And so this is going to be your right eye. So the medial rectus here is going to allow you to move the eye like this way, okay? Looking at the left. Because when the muscle pulls backwards, it will rotate the eyeball this way, right? And this one, the lateral rectus, will allow the eyeball to look the other way. You have one at the top that allows you to look up, and then the inferior will allow you to look down. Okay, what about these ones? Okay, this one, it says inferior oblique. Oblique means like diagonal, right? So these ones are obvious, like up, down, left, right. What about this one? What do you think the oblique will do? What kind of motion would your eyes be doing when the inferior oblique pulls? When my kids do that motion with their eyeball, they get a little lesson from me. Tell them not to do it. That's right, it rotates it. When you roll your eyes, right? Like, oh, come on, really? Like roll the eyes. That's the inferior oblique doing its thing. All right, so like you have a lot of muscles dedicated to eye movement, and these eye muscles have their own cranial nerve. They're dedicated for their movement. Okay. The most anterior portion of the sclera is the cornea, which is this little dome-shaped thing that we labeled last time, remember? And here is just like a quick 3D eye that I wanted to show you as we are talking about these things. Am I sharing my whole screen? You can see the eye, right? Let me just double check here.
Yep.
Oh, okay, thank you. Yeah, okay. So over here, right, you see, that's the cornea at the front, right here. It's a dome shape, a transparent part of the eye. and it's convex, okay, CONVEX. Convex means it bulges out and it allows the eye to collect light from a wide range of angle. Okay, and there's no blood vessels in the cornea, which makes sense because otherwise we will see our own blood vessels in our field of view, right? So it has to be transparent. So that is the cornea right here. Right? Now this was a thing for a while. I don't know if you guys remember reading this. I took this clip from 2017, so that's not even that long ago, like 6 years ago. Like apparently it was a thing to tattoo your eyeballs. Right? I don't know if you ever heard of this before, right? They actually try to do what's called like scleral tattoo. Right? So they inject some ink underneath the sclera, and then I don't know, you can put like a star there or something or whatever you want. And needless to say, that was a horrible, horrible idea. I can't believe people would actually try to do that. And there were countless news articles like this one where it went horribly wrong and then the person went blind after. Go figure, right? And then the FDA actually stepped in and completely ban this procedure. In Canada, of course, do the same thing as well. And you know, nowadays, if well, not nowadays, like even before this thing was invented, if you want to have a different eye color, you know, you can just wear contact lens, right? There's no need to put like tattooed ink in your sclera. But that was a thing, okay, in case you didn't know, it existed. So beneath the sclera, we have the choroid. And choroid is the vascular layer of the eyeball. Okay, that is the layer that's gonna have the blood vessels, and it's gonna be supplying the eye with nutrients, taking away metabolic waste. Okay, and at the front, the choroid becomes the color part of the eye, the iris. Okay, so your iris color is genetically determined. There are a variety of eye color. Some are more common than others. Okay, like I think mine is like dark brown. Okay, or as they call it in this picture, chestnut brown. Okay, I've seen people with blue eyes. I don't think I met anyone with green eyes before. Okay, I'm sure they exist, like seen it on TV, but I don't personally know anyone with green eyes. You might, okay, but the eye color, the pigment that determines the eye color is melanin, which is the same pigment that determines your skin color, your hair color. So that's why people of certain skin color and hair color would usually have a particular eye color associated with it, because the amount of pigment that you generate is genetically encoded. Now, there is another thing that people can do, I read about. If you don't like your not don't like, but like if you want to have like a different eye color for whatever reason, you can do what's called like laser bleaching. Okay, so using laser, they could take away some of the pigment and make the eye color a little bit lighter. You can't go the other way though. Okay, so if you have a lighter eye color and you want to have like a darker color, your only option is to wear contact lens. So the iris is not just to make your eyes look pretty. The iris is actually muscle, right? Smooth muscle. We cannot control it ourselves voluntarily. But when it constricts, it's gonna cause your pupil to become smaller, so less amount of light will go into your eye. And when it relaxes, it will open up and let more light come in. Okay, it's like taking a picture, right? If you use like old school camera, or even like with your phone, right? If you point it to something that's really bright and take a picture, it gets overexposure, right? Too much light is entering the camera. You cannot, you know, see the surrounding very well. So you have to close the aperture. You have to reduce the amount of light that's going into the camera in order for you to get a nice picture, right? So that's the same concept here with our pupil. Now, did you know, just like a fun story that you know, I was telling my kids, my daughter was asking me, you know, how come pirates, they wear eye patches? And so I was telling her, well, that's because when the pirate is fighting, right, on this pirate ship, and let's say it was a bright sunny day, and then they're fighting, and then all of a sudden they fell beneath the deck of the ship, and then they're in this dark place. Okay, so their eyes cannot see because it's so dark. So what happens is, because they have an eye patch, they can take the eye patch off and then switch it to the other eye, right? And then the other eye that was underneath the eye patch was in the dark for so long that the pupil is dilated. So after they switch the eye patch, they could immediately see in the dark environment. So, you know, that's the reason they wear an eye patch. And, you know, it's kind of true, like, because like your eye pupils are not like synchronized, okay? Like it's possible for one eye pupil to be dilated and then the other one to be constricted, okay? So, you know, that's my theory of why they wear an eye patch, but it might not be true. Anyways, so that's the choroid. Beneath the choroid, we have the retina. The retina is the deepest layer of the eyeball, and that is where you will find the photoreceptors. Okay, photoreceptors. And there are two types of photoreceptors, I told you. There are the rod cells, which are responsible for night vision as well as peripheral vision. So night vision is easy to understand. Okay, like you wake up in the middle of the night and then everything is kind of grayscale. There's not like color to it, okay? Because your cone cells, which are the ones that detects color, they're not very active at night, okay? It's mostly your rod cells that are doing the vision for you, okay? Now, the rod cells, they require a special molecule called rhodopsin for them to work. And to make those molecules, those proteins, you actually need things like beta carotene, right? Where do we find beta carotene? You must have heard people say this before, right? It's almost like an old wives' tale, right? Like what kind of food is good for your eyes? Anybody ever heard that before?
Yep, like the carrots.
Yeah, the carrots, right? The orangey stuff, right? So the one reason is because they have beta carotenes, right? As well as vitamin A, and those are the necessary raw material to make the rhodopsin that I was talking about. In some parts of the world, there are like particularly like poor countries, developing countries, they don't have access to vitamin A. And if you were in my class, we talked about this in the biotechnology lesson. Remember that people who have vitamin A deficiency, they're not able to make enough of that protein that's necessary for night vision, and so they have nocturnal blindness. Okay, they cannot see at night, like literally can't see. Okay, but when it's day, it's okay, right? And that's why people created the golden rice. Okay, if you haven't heard that before, you can look it up. It's genetically modified rice that contains the vitamin A in the grains, and so that gives access to the vitamin for people who otherwise would not be able to gain access to it. Anyways, going back to this, rod cells are important for night vision and peripheral vision. Okay, what is peripheral vision? Okay, let's do an experiment. Again, I don't know if you're watching me on your phone or on your computer screen. This works better if you're on your computer, okay? And assuming you can see me on the top right corner of your screen, okay? If your window is like somewhere like elsewhere, right, for my picture, like try to move me to the top right corner, all right? So this is what we're gonna do. Right? You're gonna read the words on the screen, okay? You just read them as I'm talking. And then as you're reading the words, okay, do you see, don't, without looking at my picture, okay, keep reading the words. Do you see that I got my hand up? Okay, you probably see that, okay? But can you see how many fingers I'm holding up without looking at my picture? Okay, if you notice my hand went up, how many fingers am I holding? Are you able to see that? Right?
No.
No, but were you aware that my hand went up? Did you guys see that my hand went up at all?
I saw some motion, but I'm not sure.
Okay, yeah, good, right? So you saw some motion, right? Okay, probably it's because like my picture is too small, right? But it's good, right? What I mean is, I am on your peripheral vision. If you're reading the words on the screen, then I am part of your peripheral vision. We cannot make out details about a peripheral vision, right? Like some of you might saw my hand went up. Some of you might saw, oh, something is moving. Okay, but I don't know what he put up. Did he put up his hand? Did he put up like a ruler, his legs, right? What did he put up, right? So like you're not able to make out details about it. Okay. Similarly, if you're looking at my picture right now, okay, then you can see clearly, you know, I put up 4 fingers, 2 on each, and then the words on the PowerPoint slides will be part of your peripheral vision. You're like, oh, I know there are words there, but I cannot read it. Okay, I cannot read it. It's on my peripheral vision. Do you understand? I have a question. Is this when a person say, I saw you in my saya? Yeah, like like sometimes like something just flashed by, right, on the side of your eye, right? Essentially your peripheral vision is picking it up. Okay, so peripheral vision is picked up by rod cells, and the key thing to know, the point I'm trying to make here is things that falls on your peripheral vision, we don't have details about it. Okay, we must shift our focus to something specifically in order to create a sharp image of that object. Okay, more on that later. So here the yellow stuff, that's the cone cell, and then the rod cells with these ones. Okay, and you can see there are a lot more rod cells than cone cells. Because the cone cells operates in bright light environment, right? You don't need a lot of stimulation. Rod cells, you need to have more receptors in order to kind of amplify the signal when it's dark. Okay. Now this is fun, okay? Well, I think it's fun. You can tell me if it's fun or not afterwards. Did you know we only have 3 kinds of cone cells, even though we can see so many different colors, right? Okay, even though we can see like orange, you know, turquoise, right? But we don't have like an orange receptor. We don't have a turquoise receptor. We only have 3 receptors. Okay. We have a red cone cell, we have green cone cells, as well as blue cone cells. Right. If you have one of those like old computer screen, like the really big one before they got the flat screen, do you remember like maybe wiping the surface with a wet cloth or like sneezing on it, and then got those little droplets on it, right? Then you can see the individual pixels, right? It was only 3 colors. It's always red, green, and blue, right? And it's actually a combination of these 3 colors that allows us to see everything in between. Okay, this is let me see if I can do that. Haven't done this thing in a while. Let's see if it still works. Let me just draw like whatever shape. Okay, let's draw a star here. Okay, and I want to do this. Okay, look. See, it's just 3 colors, right? I don't know if you can see these words. It's really small, but it's red, it's green, it's blue, and right now this thing is blue. Okay, let me turn off red completely. Okay, if I turn off red completely and green completely, then I get this very blue star, right? Okay, so let's see what happens if I turn up red as well. If I crank up red to 196, just like blue, okay? The numbers doesn't really matter, right? Just like turn it on and off. If I turn it on as well as blue, what color do I get? Blue and red together. What color do we get? You guys know? No? Well, blue and red together give me this magenta, we call it, okay? Magenta color. All right. And then we can also try to play around with this a little bit more. Okay, what if I turn up the green, okay, and then I turn down the blue? Okay, just green and red. Well, that's not working really. Hold on, something is not right. Okay, I'm not sure why it's not working. But anyway, just go back to this, maybe this is probably easier. When we mix the things together, right, we end up having the color in between, right? So with the red and the blue, we got the magenta, right? Okay. With the red and the green, you're supposed to get yellow. I'm not sure why it didn't work earlier. I'm gonna have a look at that later. Okay. And then when it's blue and green, you get this cyan color. Okay. And you can actually have more red and less green, and that will allow you to see like orange. Okay, so it's just a combination of how much you stimulate the red, how much you stimulate the green, and how much you stimulate the blue. So to see the color white, you need the red and the green and the blue all being stimulated at the same time. All right. So how do you see black? Right? What is the color black? How does it relate to the red, green, and blue cones? What do you think? Which one do we need to turn on, huh? No?
To get black?
Yeah, to get black. How do we get black? Which one do we turn on? That's my question, I guess.
I mean, they're all turned on, aren't they? Like, I mean, like add or take away?
Oh, sorry. Yeah, turned on means like activate. Okay, so like when I that's a good question. I think I was not being very clear. Let me try that again. To see the color red. Okay. Okay. Sorry. Let's start again. I'll just tell you the answer. When it's black, when there's no color, none of the cone cells are turned on. Okay, they're not activated. When I say turn on, I mean like, you know, sending action potential to your brain, right? Okay, so when it's black, nothing is turned on. How do you see red? When you see red, then you need to turn on the red cone cells, right? Okay, when you see green, you need to turn on the green cone cells and leave the blue alone. What if you want to see yellow? If you want to see yellow, then you have to turn on red and turn on green. You have to activate those cone cells, right? So when the brain, when your occipital lobe is receiving signals from the red cone cells and the green cone cells, then you interpret that as yellow. Does that make more sense? Okay. All right, let's do an experiment, okay? Hopefully it will make more sense after. Right? So what you have to do is this, okay? Look at this screen, okay? Just stare at it, all right? Don't look at me, just nothing else. Just look at your screen, your computer screen, your phone screen. Just stare at the red for a very, very long time, okay? I'm just gonna keep on rambling on as you stare at the red screen. How are you seeing this red screen? Well, you're seeing this red screen, well, first of all, because it's red, and your red cone cells are picking up this red light, okay? And then it's sending this signal to your occipital lobe, and then you're perceiving this as red, okay? Now I'm gonna switch to the next screen. Keep looking at it, and I want you to blink your eyes a couple of times, okay? What color do you see? Okay. You can type it out, you can unmute and tell me, what color do you see on this screen now when you blink your eyes?
Blue?
Yeah, like a light blue, right? Like this blue, right? Cyan, am I correct? Okay.
Yep.
Yeah. Everyone else, and does this work for anyone else? Okay. I got one thumbs up. Okay, that I'll take that as a win. All right. So what happened, right? Well, you were looking at the red screen, and then only your red cone cells are being stimulated. Okay, your blue and green are not doing anything. When you switch to the white screen, remember, how do we see the color white? Well, let's look at the color wheel. To see the color white, I need the red, the green, and the blue to be all firing at the same time, to be all turned on. But on the red screen, you tire out your red cone cells. So when you need them on the white screen, the red cones are like, oh, I am overstimulated from the previous slide. I can't do this. Okay, so it wasn't giving as much of a signal when it should have been. So only the green and the blue were sending the signal to your brain on a white screen. That's why you see the cyan. Okay. Min says, do you have to use your rod cells to see black? No, rod cell is just like in a low light setting. It doesn't do any color. To see the color black, like it's just that your cone cell, an absence of signal to the brain is how you register the color black, basically. Do you guys understand why you see the cyan? Well. Let's go to this thing here. All right. This is application number 1. You stare at a bright red screen for 30 seconds. We did that already. What color will you see if the screen is switched to the following color? You know what? Let's change it. Let's not do red here. Okay. Let's say you stare at a bright green screen. Okay. So what color will you see when you switch to a white screen? Okay, I'm gonna give you some choices, and then you're gonna tell me. Okay, are you gonna see white? Are you gonna see green? Are you gonna see yellow? Are you gonna see magenta? What are you gonna see when you switch to a white screen? Okay, well, thank you. I got my usual 5 or 6 response. And is it yellow? Well, let's take a look at the color wheel. If you're looking at a green screen, then you will tire out the green. Okay. And when you switch to a white screen, the green is not really working, so only the red and the blue are working, and you will end up seeing magenta. Okay, you will end up seeing magenta. All right, so the correct answer is actually D. Maybe ask me some questions so I know where the confusion is, because I guarantee you're gonna see those questions on your test. What's the confusion here?
I do have I'm very confused.
Okay,
So
sure.
when we are looking at a green screen for too long,
Mhm.
isn't that our green cone cells are stimulated?
Yes, they have been overstimulated.
Oh, overstimulated.
Yeah, so like you
okay.
have been using it for so long.
Long.
So when you go to the white screen, when you need the green again, they are
Oh,
tired. They're tired. They cannot send another signal again. So that's why
yeah,
only the red
understand.
and the blue will be
Oh,
sending.
because we have, when we have to see why we have to turn on all the 3 cone cells.
Exactly.
But
To
the
see white, you
green
need
is
all
too
3.
tired.
Exactly.
And
And
the
why
only-
was it too tired? Because we overstimulated
Okay,
from
I
looking at too
understand.
much green.
Thank you.
Yeah. Anyone else? Okay.
So then, Professor, if
Mhm.
in order to see yellow,
Yeah.
you have to turn it on to what, red?
You have to turn on what? Sorry.
So in order to see from green to see yellow,
In order to see the color yellow,
yeah,
you would need the red and the green to be working together.
okay.
Okay. So let's try this again, guys. Okay, listen. You are staring at a green screen. Okay, so same thing. You're staring at a green screen for a very long time. Okay, 1 minute. Then I switch to a yellow screen. I switch to a yellow screen. What color will you see on the yellow screen now? Okay. Just think about it. You don't have to tell me. I will tell you the answer in a bit. You're staring a green screen for a long time, then you switch to a yellow screen. What color will you register now?
Black?
Why black? I want to know the reason.
Well, I mean, if you're staring at green, but then it kind of turns off, but yellow is on the spectrum of green, then you turned off both. Oh, wait, no, you still turned on. So it would be blue then.
Interesting.
Red.
Okay, yeah, red is the correct answer. Okay, so let's try to explain this again. All right, so look, yellow, right? Yellow what do we need to see yellow? We need the red and the green, yes? Okay. When you were staring at the green screen, which one got tired? Which one got overstimulated? The green, right? So now the green is not really working. So on a yellow screen, rather than seeing yellow, you will just see the red. Does that make sense, Leah?
Yeah, sorry, I was just
Yeah.
overthinking that
Oh,
part.
no, yeah, it's all right. All right, let's try another one. Okay. Let's go back to the thing over here. Okay, so green screen, yellow. Oh, we did that already. Maybe just let me just make up another one. Okay. What if you were staring at a yellow screen for a very long time, and then you switch to a blue screen? Okay, switch from yellow to blue. What will you see now? Yellow to blue. What will you see? Yellow to blue. Okay, I have some people say white. Okay, I have someone say blue. I see that this is still confusing for some people. Okay, let's do this one more time. How do we see yellow? To see yellow, we need red plus green. Do we all agree on this? To see yellow, we need red and green, right? How do we see the color blue? Blue. How do we see the color blue? Which kind of cone cells do I need?
Blue cone cell.
Yeah, just blue cone cells, right? So it doesn't matter that the red and the green got really tired. I don't even need them when I look at blue, right? So on the blue screen, you will still see blue, right? Javon, Claire, does that make sense? Yeah. Okay, one more, and then we're gonna move on. What about yellow to white? Yellow to white. All right, we're getting somewhere now. Okay, so yellow, to see yellow, I need red and green, right? And then how do we see white? To see white, we need red, green, and blue. Right? This is the way you can do it on the test, okay? And you're like, oh, red and green, they are so tired. So when I need them on white, I cross them out, and it will be blue. Okay? Are we crystal clear now? If I give you this question on the quiz, everybody's gonna get it right? Okay. All right, let's move on. So this is another one, but you know, we spent so much time on the optical illusion already. We're not gonna do it. Okay, you can play with this yourself. You just stare at the blue dot in the center for like a 30 seconds or something, and then you switch to this black and white photo. But your brain will paint it for you. Okay, you will actually see color, even though it's just grayscale. Okay, so you stare at this, and then you switch to this. It's kind of cool. You can try that on your own. All right, we're gonna move on. Color blindness. Okay, color blindness. This is like a common test, right? The Ishihara test that is used to see if someone has color blindness. I mean, there are many different forms of color blindness, but the most common kind is the blue-green color blindness, where the person is not able to distinguish shades of red and green. So what numbers do you guys see? Okay. Do you wanna? Brill asked, do you want us to memorize the color combination for the test? I mean, like, you don't have to memorize it per se, but like it's good to like have a reference here, right? Like maybe on your phone or print it out. Because you definitely need to have this in order to answer some of the questions. Okay. Okay, yeah, I'm seeing a lot of 74. Anybody not see 74 here? Okay, like, I mean, you don't have to tell me, but if you don't see 74, you should go see a doctor. Okay, unless you already know that you're colorblind. If for a colorblind person, they will not see 74. They will see, I forgot what they will see. Let me see my note. They will see 21. Okay, they will see the number 21. So if you see 21 here and you didn't know you were colorblind, right? Then you should see your optometrist. Okay, so that's the Ishihara test. I think that's how you pronounced it. It's an X-linked condition, as you learned last semester, right? It's passed on through the X chromosome. Males are more likely to get it than female because female has 2 X's, right? Okay, this is a sex-linked recessive, X-linked recessive. Okay, moving on. The retina. Okay, so in the retina, there is an area called the macula. Okay, it's at the center portion of the retina. Let me see if I can pull the 3D eye here in again for you. Okay, right there at the back, in the center, right? This, this darker shade area, that whole thing is called the macula. And at the center of the macula is the fovea. Okay, fovea. We labeled the fovea last time. Okay? The fovea is like a little pit at the inside of the macula that contains the highest density of cone cells. Okay, so that allows us to create the sharpest image of whatever we are looking at, right? So if you're reading the words right now on the screen, those words are going to be projected onto your fovea. Everything else, okay, like me in the little picture at the corner of your screen, right? Your keyboard, maybe your notes, all those things are going to be on your peripheral vision, okay? And while we are aware of some information of those objects, we cannot make out details about it, okay? We can only focus on one thing at a time. We can only have one object of interest on our fovea at any given moment, okay? Everything else will have to be in the peripheral vision. Now, your eye doctor sometimes will take pictures of your eye and it will look like this. Okay, so they will see if your macula is in good shape. The fovea again is like kind of in the center of it, and then a little bit to the side of it, that's where you will have your blind spot. Okay, your blind spot is where the optic nerve exits the eyeball, right? Yeah, let me see if I can show you in this diagram again over here. Right, so the doctor will take a picture through the pupil, and we saw the macula, the fovea, and over here where the optic nerve is exiting this location, right? That is your blind spot. There are no photoreceptor on it because the nerve is in the way, right? And you can see there are a lot of blood vessels that are also coming out from this area as well, right? To go into your choroid. Okay, so that's the blind spot, also known as the optic disc. So let's try to find our blind spot. Again, this probably works better if you are on the computer screen. Phone, maybe, I don't know, we'll find out. Okay, so this is what you have to do, right? Just follow my instruction. I want you to close your left eye. Okay, you can cover it up. Close your left eye and then stare at the circle. Stare at the circle. Okay, just stare at it. And then you move your, okay, so at this point, before you move, when you stare at the circle, you should still be able to see the plus sign. Okay, so the plus sign is on your peripheral vision. Huh? So keep staring at the circle and then you move your head closer and closer to the screen. There should be a point where the plus disappear all of a sudden. Okay, try that. So cover your left eye, stare at the circle, move your head slowly towards the screen, and then there is a point where the plus suddenly disappear. Let me know if it works, okay?
Yeah.
Yeah.
Yes.
But
Okay.
when my eye is almost touching the screen.
Oh, Okay. Yeah, so you it depends how big your screen is, right? And it depends on how big the picture is. Yeah. And if you cover your eye initially and the plus is already gone, that means you're already at your blind spot. Okay, so that location where the plus disappear, that's where the blind spot is for the right eye. Okay, you can do the same thing with the left eye. You cover your right eye, you stare at the plus, and then you move towards the screen slowly, and then at some point, the circle should disappear. Okay, so each eye has a blind spot, but when you open up both eyes, right, you will be compensating for each eye's blind spot, right? Like you can move in and out with both eyes open. It doesn't matter how close you get or how far you are, you will always be able to see both of them. Okay, because again, the blind spot of the left eye is compensated by the right eye and vice versa. Okay. All right, we're just gonna skip some of these questions because we still have a lot to cover. The cornea is part of the, and remember cornea is the convex thing at the front. That would be the sclera. 3 types of cone cells. Maybe we should try this one. 3 types of cone cells. You should know this one, right? We spent like a good 10 minutes on this thing. Yeah, okay, good. Right. A lot of people answered. The answer is D, right? Red, green, and blue. Excellent. Which of the following controls the size of the pupil? Control the size of the pupil. That's right, the iris. Good. The sharpest image is projected onto the fovea, okay? And the fovea is located in the macula. If you're a little bit confused again, look, this is the eyeball. Okay, this is the optic nerve. So that's the blind spot. Blind spot. And then just next to it, you would have an area called the macula. In the middle of the macula, there is a little pit, and that's where the fovea is. Okay. Think of this as like a target, like a target. Right? The whole thing, the whole target is called macula, and then the center of it is called the fovea. Okay, more stories. Lens is crystalline structure located behind the iris and the pupil. That's pretty obvious. And I told you the kind of lens that we have in the eye is called a converging lens. A converging lens causes light rays to come together like a magnifying glass. A magnifying glass is converging lens. The opposite of a converging lens is a diverging lens. It spreads the light out. Right? So the headlight of a car, right? There is a converging lens at the front that allows the beam to spread out, right? So you can illuminate a wider area, right? Think about like a searchlight in the lighthouse. There is a diverging lens. You want them to spread out. Okay? But for the eye, we want to use a converging lens to focus the light rays onto the fovea. I should probably write that here. Focuses, is that how you spell it? Focuses light rays onto the fovea. Okay, onto the fovea. The shape of the lens, okay, more specifically, we could say the thickness. Okay, the thickness of the lens is controlled by suspensory ligaments and ciliary body. Okay, so we have these suspensory ligament that goes around the eye. Remember, this is a mid-sagittal cut, so you don't really see it all the way around. But if you were to look at it from the front, the suspensory ligaments would go all around. Okay, kind of like, you know, drawing a sun when we were a kid, right? It goes all the way around. And then on the other end, it would be connected to these ciliary body or ciliary muscle. Right? For us, it's the same thing. Right? We're not going to make a distinction. Okay. So please remember this: far away, to see something that are far away, you need the lens to be thin, thinner lens. Nearby stuff, you would need a thicker lens. Okay, so imagine you are in like a regular classroom. Okay, like, you know, last semester, maybe you were in a regular classroom. When you look up from your notes and you look at the projector screen, then your lens needs to be thinner. Okay, otherwise you won't be able to see the words. And then when you're writing in your notes, okay, then the lens needs to be thicker. Right, so the switch must happen quite quickly for us to maintain focus. The ability to change the thickness of the lens and to maintain focus as we shift our vision from like far away to nearby, this ability is called visual accommodation. Visual accommodation. Okay. So there are other things that goes on in visual accommodation, but we're just going to focus on the lens, okay, to keep it simple. So let me try to explain this as best as I can. My hand here represents the ciliary muscle. Okay, it's circular, and so when it contracts, they grab onto each other, and it actually makes like a smaller circle. Okay, and then when they relax, it opens up. Okay, like a loose analogy to help you like kind of understand it, because some people have trouble understanding why is contracting making it smaller. Like they have a hard time understanding this. Okay, so think about, like, you know, drying a towel, right? If you contract it, right, and you twist it all up, it becomes smaller, right? And then when you open it up, it becomes bigger when it's relaxed. Right? So that's kind of like a loose analogy for you to remember. So what happens is, you know, think of the rubber band. Oh, you cannot really see it right from my screen. Oh, you can kind of see it. Okay, so this is a rubber band, and if the ciliary muscle relax, then the rubber band is not being pulled on. But sorry, let me try that again. The rubber band represent the suspensory ligament. So when the muscle contracts and becomes smaller, then the rubber band has no tension. But when the muscle relaxes and open up, then there is a lot of tension in the suspensory ligament, right? In the rubber band. And that's kind of what happens here. Okay? When you're looking at something that's far away, the muscle relaxes and it opens up, it gets bigger. And when it gets bigger, it pulls on all the suspensory ligaments and causes it to become tight. And because the lens is being pulled in all directions, the lens becomes thinner. On the other hand, when you try to focus on something that's nearby, the ciliary muscle contracts, and then it causes the ligaments to become loose, right? And now the lens is going to not spread out as much, and it becomes thicker. And that's what you need for this. So this is like reading a book. Okay, this is like trying to read the bus number as is coming to the bus stop. Okay. Now, as we age, our suspensory ligament is gonna lose its elasticity, like the rubber band, right? If I keep on stretching it, like many times, if I do it over, you know, couple thousand times, I guess, what happens is the elastic band eventually will lose its elasticity. Okay, like you stretch it out and it might not go back to original shape. And that's the same thing with our eyes, with the suspensory ligaments. So when we get older, we lose the ability to do visual accommodation. It doesn't happen as quickly. So we have what's called presbyopia. Okay, presbyopia, right? And what happens is like you try to read something that's close by, but you won't be able to see the words. Like I'm surprised, like I'm like 40 and I'm already seeing my vision problem with the presbyopia. I was trying to read the ingredients on like a pill bottle the other day, right? And I wasn't able to see it. Okay, like I actually had to try to put it further away, closer, and like nothing works. In the end, I just use a magnifying glass and I was able to see it. Okay, but the reason is because the suspensory ligaments is not able to change the shape quickly. It's not able to cause the lens to become thick enough to help you maintain the focus. So what's another option for people with presbyopia? What do they need in order to see clearly?
Eyeglasses.
Glasses.
Yeah, but not just like regular glasses, right? They have a
Reading glasses.
yeah, it's called reading glasses, right? Yeah, exactly. Okay, and for people who also have like farsightedness or nearsightedness, right? Then they would sometimes wear like bifocal glasses, right? So like the top part of their glasses is to correct their regular farsightedness, nearsightedness, but then the bottom part is to correct for the presbyopia. Laser, I don't think actually will correct this problem because this problem is not related to the curvature of the lens, which is what the laser corrects. This has to do with more like the muscle and the ligaments not able to change the shape quickly. So I think reading glasses is the key thing here. Okay, moving on. The lens divides the eye into two compartments. We know that there is the anterior compartment, there's the posterior compartment. And at the front here, we have the aqueous humor. Okay, so this part is filled up with the aqueous humor. I'm just gonna put AH here, aqueous humor. And then the back here is filled with the vitreous humor. The aqueous humor provides nutrients for the cornea as well as the lens because those are the parts that don't have the blood vessels, okay? And they will need to have some source of nourishment. That's what the AH is for, aqueous humor. And the vitreous humor is just to reinforce the shape of the eyeball, okay? So it doesn't like, collapse under its own weight. Right, so those are the two things. Couple things that could happen here. Floaters. For people with bad eyesight, floater is a more prominent problem. What is floaters? Well, first of all, if you have it, you already know what this is. Okay, for those of you who don't have it, again, congratulations. But let me try to describe to you what it's like. Okay? So people with floaters, when they look at a bright screen, like a bright white computer screen or a bright blue sky, and they blink, and they will see these things kind of flying around in their field of view. Okay, and they kind of look like this. Okay, so these are actually debris that has fallen off from your lens, and they have nowhere to go. They're too big to be drained in the dural sinus. In the sclerovenous sinus, and they are just stuck there in the aqueous humor, right? In Cantonese, we call this like the flying mosquito syndrome, okay? Because they look like little annoying flies that are buzzing around in your field of view. Okay, so nothing much you can do with it. Like I think laser might be able to fix some of it. There might be some eye drops that would help break them down into smaller pieces, but for the most part, they're just like a nuisance. Okay, so that's floaters, protein debris in your aqueous humor. Cataracts. Cataracts is what happens when the lens turns cloudy. Okay, and when it's cloudy, the light cannot pass through. you can't see anything, right? Okay. Fortunately, we now have the technology to replace your lens. Okay. Artificial lens. It used to be you have to like take a lens from a donor, but now you can use artificial lens, and it will restore your vision. My mom just had her cataract surgery done like 2 months ago. Okay. And her nearsightedness is gone as well afterwards. Okay, because you replaced the lens, right? And lens is part of the problem with the nearsightedness. So once you got the new lens, even the nearsightedness is gone. So that was a added bonus for her. Next we have glaucoma. Okay, glaucoma is when the aqueous humor is not draining. Okay, it's not draining fast enough, so the eye pressure increase. Right, so some of the excess aqueous humor is going to go to the vitreous humor, and it's going to be increasing the eye pressure, which in turns, the high pressure in turns compresses the optic nerve and damage it over time. Right, so in glaucoma, the person would slowly lose peripheral vision, and then it will be blind completely towards the end. There are no cures for glaucoma, but there are treatments to slow down the progression. And the treatment aims at reducing the eye pressure, right, so that it's not going to cause further damage. But it's like a progressive thing that could eventually result in complete blindness. All right, so those are some eye conditions that I wanted to tell you about. Okay, pathway to brain. This one is confusing. Okay, let's try to do this. Can we pull out the study guide and go to this diagram? And if you don't have the study guide, that's okay. You can just quickly draw something like that, huh? So first of all, what are we looking at? This is a top view, okay, of a transverse section. So this is the right eye. And over here, this is the left eye. All right. So things that are on your right side, we call that the right field of view. It's like a tongue twister. It's hard to say. I'm just gonna put like a purple person here. Okay. That is right field of view. Field of view. And then we're gonna have a blue person here, and that would be the left field of view. Now, it's not exclusive, okay? Like it's not like everything must either be on your right field or left field. There is like overlapping in the middle, right? Okay. Some things are like exclusively on the right field. If you have something that's like all the way out here, then this is exclusively on the right field. But if you have something that's like in the middle, like a green person in the middle, then, you know, it's part of the right field as well as part of the left field, right? But to keep things simple, we're just gonna look at the right field as if it's on its own, and then the left field as if it's on its own. All right, the pupil is out here at the front. Okay, that's where the light enters. Now tell me, which half of the eyeball will the left field of view be projecting on? Okay. Let me repeat. Sorry, go ahead.
Left eye.
Sorry, I didn't hear you.
Left eye.
Left eye. Okay.
Mm.
So let me rephrase my question is so that it's a little bit more clear. Okay. The left field of view, right? When light enters the eye, which half of the eyeball will the field be projected on?
The right side.
That's right. Okay. for both eyes, right, Tashana? Right, for both eyes. So let's see, the light is gonna go into the right eye, okay, through the pupil, and it's gonna go into this eye as well. And the right half of the eyes is responsible for perceiving the light from the left field. Okay. Just gonna pause for a second and see if there are any follow-up question that needs to be clarified. Okay. So what happens next? What happens next is this. The signal from the cone cells and the rod cells in the right half, they're gonna travel. Right? These are action potential where they're gonna travel along the optic nerve. Okay, so that's the left optic nerve. You guys remember the optic nerve, right? That's the thing that comes out from the back of the eyeball. Remember all the eyeball pictures we have? There is a thing that sticks out at the end, right? That is the optic nerve. So that's the left optic nerve. And over here, that's the right optic nerve. Okay. So they're gonna travel all the way to the middle over here. And that is the optic chiasma that we mentioned before. So let's take a look at this path first, okay? So the right optic nerve containing the left FOV information will actually continue on the same side. Okay, so we call that ipsilateral, right? You might remember learning that last semester. But over here, for the left eye, it will cross the middle. It will cross the middle at the optic chiasma and merge with the other nerve and continue onward. Once you pass the optic chiasma, we are inside the brain. We are inside the central nervous system. So we now call this the right optic tract. Right? That will go to the right thalamus. And then the information will be projected onto the right occipital lobe. So your entire left field of view is going to be processed by the right half of the brain. Okay? Even though the information is actually coming from both eyeballs. Okay? But they join together after the optic chiasma, and then it will get sent to the right occipital lobe. Okay? So right lobe is responsible for processing left field of view. Okay, let's take a look at the other path. Over here, the right field would enter both eyeballs, but it will get projected on the left half of the eyeballs. And then the left optic nerve at the optic chiasma will join with the optic nerve from the other side, and then it will continue as one on the left optic tract. Left optic tract. And then it will go to the left thalamus. And then it will get projected onto the left occipital lobe. This is something that you have to draw out a few times yourself before you can, make sense of it all. Okay. So the left occipital lobe will be processing the right field of view. And you probably have some questions, but just hang on to them for now. Let's go through some questions here and see if it will help you out. So let's see. The right optic nerve carries information from which field of view? Okay, so optic nerve, that's before the chiasma. After the chiasma is a tract, right? So over here, right optic nerve. Let's take a look. So right optic nerve, well, I see two colors. Blue represent left field of view, right, pink represent right field of view. So the right optic nerve contains information from both. Okay, both field of view. Just gotta use a different color here, so it's not gonna get confused with the other writings. So both field of view, right? You see two colors, right? That tells you it's both. How about this one? The left optic nerve carries information from which field of view? Same thing. Left optic nerve, right? Two colors. Blue is left field, pink is right field, both. Okay, so that's pretty straightforward. Next, the right optic tract carries information from which FOV? Okay, so the tract is after the chiasma, right? After the crossover. Chiasma means crossing over, right? You can see that's where the fibers from each eye crosses the midline. So if you look at the right optic tract, well, we see only blue, right? So that's left FOV only. And for the left tract over here, it's right FOV only. Okay, how about this one? The right optic tract carries information from which part of the eye? Right optic tract is right here. And if you trace it all the way back to the eyeballs, it's the right half of the eyeballs, both eyes, okay? And on the other hand, the left optic tract, this one would be carrying information from left half of the eyes. Okay. So if someone damage, let's say the right optic nerve here, okay, if we damage this here, what's gonna happen? Well, if you damage this, then your right eye will not be able to send signal to the brain. You can still see both fields because your left eye is still functional, right? Okay. On the other hand, if you damage the right optic nerve, a tract rather, if you damage the right optic tract, right, then both eyes will still be able to send signal to the brain, but half of the eye will not be working. So if this is damaged, you will not be able to see your left side. Okay, even though your eyes are perfectly fine, right? You can see the left side. You will only be able to see the right side. Okay. Any questions before we take our break?
Yeah,
Yes, Shirley.
Professor,
Mhm.
I'm confused, like the E and F. The
E
right,
Uh-huh.
the right optic tract, carries.
Yeah, right optic tract is this one, right? So
Yeah, this one carry information
from the
from.
right half of the eye. So if we trace all the way back, the blue is the right half of the eyeball, right? Because the left
okay, yeah.
field enters the right half of the eye.
Yeah. Left view enter the right. Yeah, okay.
Right.
Thank you.
Anyone else? Okay. Yeah, let's take a break, and then we still have a few more slides for the eye, and then we're gonna move on to the ear. Okay, see you in about 5 minutes. Okay, so that was the eye field explanation, and just to show you, I did not make that up. It actually does happen. This is an like a professional drawing. I keep on calling them, right? These are textbook diagrams that illustrate the same concept. And like I said, it's probably a good idea that you draw it out a couple of times. Like I remember when I learned this for the first time, it made no sense. Right? It only started to make sense when I physically draw the whole thing from scratch, and then I can see what goes where and why. All right, we're gonna go through another note for common vision problem. Let's do a class note here, common vision problem. Eye vision problem. Okay. If you wear glasses, you either have nearsightedness or farsightedness. Okay. So what is it? What's wrong? And how do we correct it? That's what we're gonna talk about. So two types of lens. Let's just talk about that first. We have this one, right, which I already explained to you, is called a converging lens. Converging lens. Okay, or sometimes called convex lens because it bulges out. And what that does is it takes light and then it focuses it, right? That's one type of lens. That's the one that we have in the eye. Okay. The other kind is diverging lens, which spreads it out. Diverging lens. Or concave lens, because it caves in the middle. Okay. Just gonna search up a picture here. All right. So the situation with the eye is like this. Oops, sorry. Oh, that didn't copy over. Give me 1 second. That was a math equation from math class, but I wanted to copy this instead. There we go. Do you guys know what this is? Like, if you are maybe a little bit older, you might remember seeing this thing at school. This is
Is that
an
a projector?
yes. This is over. When I first started teaching, we had no like fancy like computer projected screen, and I would have to use this thing to teach. We have those transparencies that we put on top, and then I would write on it, and this thing's like produce a very bright light that is just really tiring on the eye. Anyway, so that's what this is, right? So the overhead projector, you basically put your material here, right? The transparency, and then the light goes up here, and then this, reflects the light, and then it goes on to a screen. Okay, so like, if you have the letter A here, this is gonna get projected onto the screen, and then you see A. Okay. You guys have seen this, right? Some of you must have. Okay? And for those of you who haven't seen it, that's basically what it does. All right. Now, what we need to do before this works is there is a little little knob here that helps you adjust the lens. Okay, so if you move this knob, it's going to help you focus the letters on the screen. Okay, if it's too far away from the screen or too close to the screen, then this is going to appear to be like blurry, right? And you won't be able to see clearly. So we have to adjust the focus. So our eye is like that. Our eye is like that. Okay, you have your eyeball. Okay, this is going to be your optic nerve, and then this is the fovea. The fovea is like the screen. Okay, fovea is like the screen, and then you would have your lens here. Okay, so in order for you to see things clearly, you must be able to focus onto the fovea. Okay, so this is what happens in a normal situation. Okay, normal vision. Your lens is focusing on the fovea, no problem. So let's take a look at what happens when we do have problems. So nearsightedness. Fancy name for nearsightedness is myopia. Okay. First of all, this is not obvious for people who don't have eye problem. Nearsightedness is when you cannot see far. Okay. The name of the condition tells you what you can see. Okay. What you are able to see. So nearsightedness, you are able to see nearby, but not far away. Okay. And depends how bad it is, right? Far away might not be very far away. If I take off my glasses, like I can't even see what I'm writing on the screen. And you're like, really? The screen is not that far, right? But like I have really, really bad eyesight. And you know, part of it is genetics. Okay, so that's nearsightedness. What happened in nearsightedness? Okay. In nearsightedness, the eyeball is too long. Okay. This is an exaggeration, of course. Like nobody's gonna have like an egg-shaped eyeball. Okay, that's like really messed up. But like I'm drawing it to exaggerate to help you understand. Okay. So like you would have your lens here, yeah, your regular lens. Okay, and then the eyeball is too long. So that's like in our situation, that's like moving the screen further back. It's going to be blurry, right? You're not going to be able to see properly, right? So in this particular case, the light rays, they're going to come in, and then they're going to be converging too soon. Okay, converge before, in front of retina, in front of fovea. And you were supposed to be right here on the screen, but now is here instead because we move the screen back, right? Originally, the eyeball is supposed to be like circular, right? But now it's too long. So the light rays are focusing in front of the retina. Does that make sense? So that's blurry. Now, like there is some math that involves that tells you why it only affects far away and not nearby. We're not going to go into that. Okay. So how do we correct it? How do we correct it? What do you think? We have two types of lens, right? To get glasses. Should we, to correct this problem where the light rays are coming together too early, too soon in front of the retina, should we get a converging lens or a diverging lens? Do you know what kind of lens you're wearing? Right? If you have if you wear glasses, do you know if you're wearing converging lens or diverging lens? Right? What do we need to correct nearsightedness? That's the more common form of eye problem.
Converging,
Sorry, which one?
converging.
Converging for nearsightedness, okay. Anyone else? Just wondering. Let's do a poll.
Diverging.
Diverging,
Because
yeah.
for-
Sorry, you want to?
because-
Sorry, go
Because
ahead.
you said nearsightedness doesn't have to do with your sight being near, right? Or it
No,
doesn't?
nearsightedness just means you are only able to see near stuff, right? You
Yeah,
cannot see far.
but for conversion lens, it's the light coming directly into your eye, which would be near. So the diversion would be for far, no?
Ah, interesting. Okay, I see where the logic is happening. Okay. Well, good try though. Let me try to clarify this. Look, the light rays is coming together too soon in this case, right? Because you don't want it to come together like here. You want it to come together like there, right? So what you actually want to do is you want to give it a diverging lens. You want to give it a diverging lens so that the light rays comes in through your glasses and it's going to spread it out further. It's going to spread it out first, and then your lens is your actual eye lens will cause them to come back together. So if you spread it out just enough, right, so that your actual eye lens will cause them to focus again on the fovea, right, then you corrected the problem. Okay, you don't want to overspread it because then now it will be like behind the fovea. You want to spread it just the right amount so that you can focus it back on. Does that make sense? Diverging. Okay. Now, on the other hand, the farsightedness, okay, this is hyperopia. Right? So if you're farsightedness, you are able to see far away, no problem, but you cannot see nearby. Okay. My younger daughter is farsightedness, has farsightedness. Okay. Like before she got glasses, she's not reading. Okay, she was having trouble reading. Like even though she's supposed to know how to read, right? And then we found out that she actually cannot see the words. That's why she can't read. Okay, so then we got her glasses. So what's the problem here? For the farsightedness, the eyeball is too short. Okay, again, this is exaggeration. So you have your lens here, and because it's too short, right, the light rays actually will be converging behind. Behind the retina. And again, it's blurry. So we actually need to have converging lens. Converging lens. So how does converging lens help? Well, the light rays are not coming together soon enough. So the converging lens will converge a little bit for you, and then you converge it more with your own lens. And then it's going to fall right in place. Okay. What did I say? What other lens that we encounter in daily lives is converging? Do you guys remember?
Reading,
Sorry, which one?
reading.
Oh, not the reading glasses.
Your question.
Car? The lights on the car?
The light in the car is diverging. It spreads it out so we can see like wide area, right? Remember? But thank you for trying. Converging lens, magnifying glass, that's what I meant. Okay, magnifying glass, right? It's converging lens. So people who wear glasses for farsightedness, their eyes are a little bit bigger. You probably have seen someone like that before. Now, let me show you a picture. Right? Look, which one is the diverging lens? You think the one on the right or the one on the left? Just by looking at it. I hope you can tell.
Left,
This one is diverging lens?
left is diverging.
That's right. Okay. This one is converging, right? You see the eyes are bigger, right? Okay. And also, you can tell because for people who wear, I should write down. This is diverging lens. And what do we need diverging lens for? Diverging lens is for myopia, nearsightedness. Okay, this is converging lens, which is for hyperopia, farsightedness. Okay, and you can see when the person turns their head a little bit to the side, diverging lens, people wearing diverging lens, you can see their face cave in compared to the rest of their face. You see, caves right in. But for people who wear converging lens for farsightedness, you can see their face bulges out. Okay. So now you can tell what the person is wearing. So if you are nearsighted, you will need this one. Farsighted, you will need this one. I remember seeing an episode of Survivor. I used to like that show. But now, after like 20 something season, I think I stopped a long time ago. But anyway, I remember watching an episode of Survivor. For those of you who don't know what Survivor is, like a reality TV show. A bunch of people go on like an island or something, and they have to do games and survive and win. Okay, so you're allowed to bring like one item or something, and one of the first thing that contestants usually wanted to do is to start a fire, right? So I remember this guy, he was wearing glasses, and he was trying to start a fire with his glasses, and he couldn't do it. And I remember seeing it, I was like, oh, that's so dumb, right? He's nearsighted, and so he's wearing a diverging lens. You cannot start a fire with a diverging lens, right? Because what does a diverging lens do? Diverging lens spread the lights out, right? You're not going to be able to concentrate a beam and make it hot enough to start a fire, right? Okay. So if you're going to start a fire with glasses, you would need to be someone who's farsighted, and then you have the converging lens in your glasses. Yeah, but even so, it's not guaranteed, because the curvature matters. Okay, if you have the wrong curvature, you might not be able to create a focal point that's strong enough. Okay, the curvature for magnifying glass is very different from those of eyeglasses. All right, like think about it, right? If you were actually wearing a magnifying glass type of lens, and the focal point can concentrate a beam so much, then it might actually burn your eyeballs, right? Okay, so like we don't want that. So even you're wearing the right kind of glasses to converge the beam, you might never be able to generate a beam that's strong enough to start a fire. Anyways, I just thought that was a funny story. Okay, last one is the astigmatism. Okay? Astigmatism usually accompanies both nearsighted or farsightedness. Okay, my astigmatism is also very bad, and for those of you who also have astigmatism, you would know it's worse when it's at night and raining. Okay, so astigmatism basically, instead of having a single focal point on the fovea, it spreads out all over the place. Okay, so it appears to be scattered. Right? So to describe to those of you who don't know what it's like to have astigmatism, at night when I'm driving and if it's raining, all the street lights and the traffic lights, they look like fireworks. Okay, rather than seeing like a actual concentrated red circle in the traffic light, it spreads out, right? like firework. That's why I don't even drive at night. I always tell my wife to drive. So that's astigmatism, and you would need a lens that would correct the unevenness to help refocus onto your retina. Okay, so that's that. Oh, I actually have a picture for it. Okay, normal vision, and then this is astigmatism. Okay, let's see if you learn anything from me. Go for it. Someone asked if laser can help astigmatism. I don't think so. I think laser, you can get rid of the myopia and the hyperopia, but the astigmatism will stick around. I think you can reduce it, but you cannot get rid of it completely. Not sure why though. Okay. Oh no, I accidentally deleted all the anyway, I saw various selections. Okay, it wasn't like just a single choice. So let's go through it. Common vision condition in which you can see distance object clearly, but not nearby might be blurry. Right? So remember, the names of the problems tell you what you are able to see. So if you're able to see far away, then that's gonna be farsightedness. Okay, and in farsightedness, the fancy name for it is hyperopia. Okay? Hyperopia is farsightedness. Myopia is nearsightedness. You can see near but not far. Presbyopia is when you lose the ability to accommodate as you age. So when you shift your focus from far away to nearby, like I cannot read the pill bottles, that's presbyopia. Astigmatism is the night vision thing, right? Like everything spreads out, the lights are scattered. Okay, answer is B. Ooh, here's another one. Correct answer is glaucoma. Cataracts is when the lens becomes cloudy. Floaters are those like annoying things that fly around in your aqueous humor. Glaucoma is when you progressively lose peripheral vision due to degeneration of optic nerve, and eventually you will lose the vision completely. Presbyopia, I just described to you on the previous slide, right? That's when you need reading glasses, right? You lose the elasticity in your suspensory ligaments. You can change the thickness of the lens quick enough to maintain focus. That's presbyopia. So correct answer is C. Which of the example is presbyopia? Okay, D, right? Need to hold a book far away to read it clearly. Right? You see people like try to put it away to try to see it, right? That's presbyopia. D. Oh, okay. That's
Sorry,
yeah. Sorry. Yeah.
Professor.
Mhm.
The
Presbyopia. Yeah.
yeah, presbyopia and what's the difference between presbyopia and hyperopia?
Oh, hyperopia is farsightedness. You can see far away. You cannot see nearby all the time. Okay.
All the time.
Yeah. Presbyopia, it takes time. Okay. Like if you hold a book here long enough, then you can see you can eventually see it. Okay. It's because your lens is not changing the thickness fast enough. Okay. So like eventually if you just wait for it, but sometimes the waiting takes very long time. Okay.
Okay,
Like,
thank you.
yeah. Okay, this one, right? Let's try this. Hey, don't forget to try these questions again before you do the test. Some of the test questions are these questions, right? Okay, so remember the eyeballs, right? Eyeballs, and then you have this thing, right? So right optic tract, that's right here, damaged. Okay. If you look at the diagram that we drew, that was blue. Okay? And if you trace it all the way back, the right optic tract carries visual information from the left field of view. So if you damage that, the whole left field will be gone. Correct answer is B. Okay, you just have to look at the picture that we drew and match the color. Like I said, right, don't worry if you don't completely get it now. Just, redraw it yourself, rewatch the recordings, and you should be good. Okay, that was the eye. We totally gonna have to go to 11 today. With sorry, like I said, sometimes I plan to have a short lesson, but end up going all the way. Looks like it's gonna be like that today. So hearing. The ear has two sensory functions. Many people don't know this, right? People think, oh, ear is for hearing sound. That's part of it, okay, detection of sound. But the ear is also responsible for detection of equilibrium, okay, equilibrium. And to do either of these, the type of sensory receptor that is required are called hair cells. And hair cells, if you remember, we learned it last class, they are a type of mechanoreceptor, which will detect either pressure change or body movement. Hair cells are not the only type of mechanoreceptors. You have other mechanoreceptors on your skin that detects, people poking at you, that kind of pressure. Right? These kind of pressure is different. And these are like pressure that are created within some sort of fluid, okay, as we will see. Right? Just think about like swimming. The further you go down, the higher the pressure, right? So that's the kind of pressure that we're talking about. So here is the ear. I think we should go ahead and label the ear first. All right, and then we're gonna come back to some of these stuff, and I'll explain as we go with the ear. So the ear is here. There we go. Give me a second. So the ear is separated into external ear, middle ear, and inner ear. This like the physical ear that we think about when we say ear is the external ear, and we call it has a fancy name. It's called the pinna. Pinna. Okay. It's made out of cartilage. That's why it's flexible. You can bend it. It's just gonna go back to shape. And this here is the earlobe. Right, depending on your genetic makeup, it might be attached or detached. Okay, there's no cartilage in earlobe. That's where you can put a ear ring in it. Yeah, I mean, like you can put it in the cartilage too, but I think those hurt a bit more and it takes longer for it to heal. So without cartilage, that's the common place. Sound is gonna go in to your ear through this thing. That is the tympanic, that's the auditory canal. Okay. Auditory canal. It funnels the sound wave towards the eardrum. Okay, so this stuff here, that's the external ear. Or outer ear. Right. F is pointing at this thing here, which is kind of like the interface between the external and the middle ear. I guess you can still classify it as external ear, okay? This is the eardrum, but nothing's ever easy in anatomy. This is called the tympanic membrane. The eardrum, really. Like nowadays, you can take the temperature, right? With those digital ones, you stick it in the ear, you press it. It actually sends infrared in waves to your tympanic membrane, and then it's able to check your body temperature. Okay. At the hospital, they can actually do it from your forehead as well. They have these really expensive ones. But like at the mall, like at the beginning of or like the middle of the pandemic, like at the mall, they have these things where they check your hand temperature before you can go in. That's just dumb. Okay, that's just like for show, right? Because your hand temperature is completely not reliable. Right? Think about the winter when it's really cold, right? Which part of your body gets cold first? Is your hand and feet, right? So your hand and feet by anatomy is going to be a couple of degrees colder than your actual core temperature, right? So you might as well not check it if they're like, scanning the temperature of your hand. It's just like one of the dumbest things I've seen. Anyway, so that's the tympanic membrane. And the function of tympanic membrane is to convert sound energy into movement. Okay, so when sound wave strikes the tympanic membrane, it's going to vibrate. It's actually the reverse of how a speaker works, right? If you ever look at the inside of a speaker, right? It vibrates to produce sound, to produce music. This is the other way around. The sound causes the eardrum to vibrate. Next here, next portion, we have the middle ear. Middle ear, like kids, they very common for them to get like middle ear infection, right? That's what we're talking about, this space right here, middle ear. Right, so in the middle ear, we have 3 bones. 3 bones. Okay. I will tell you the names of these bones later, but collectively they are called ossicles. All mammals have ossicles. We are mammal. And the function of ossicles is to amplify. Amplify sound. About 20 times. It's a lever system. By using a lever system. Lever, right? Like if there's a big rock here and you want to move it, then you can put like a long plank here and put a little smaller rock here. You push down here, then this goes up, right? It magnifies the force. So in a similar manner, the ossicles amplify the sound. Okay? Ossicles. And then we have this tube right here. This tube. This is called a Eustachian tube. Eustachian tube, right? Or auditory tube. And what it does is it actually connects to nasopharynx. and it's normally closed, but when you swallow, it opens up. Okay, and the function is to equalize pressure with surroundings. Okay, so like if you've been on a plane, or even if you get in an elevator that goes really high up, like your ear gets plugged, right, as you go up. That's because the pressure of the surrounding drops as we go up, and then there's too much pressure in our head, so we feel like our ears are plugged. When you swallow, it opens up the eustachian tube, lets out the extra gas, not gas, like the air, right? And then it equalizes the pressure. So this actually connects to your nasopharynx. I remember one time I was teaching this in class, and I was saying, oh, you know, like ever seen like in a movie someone smoking, and then they could puff smoke out of their ear, right? And then one guy in the class put up his hands like, I can do that. I was gonna ask him to do a demo, but you know, he was so enthusiastic to tell me that he could do it. And the reason that something like that is possible is because the eustachian tube is connected to the nasopharynx. Okay, and again, normally the air comes out to equalize the pressure, right? So it's not too far-fetched that, you know, if you smoke, the cigarette smoke can come out too. Although, of course, smoking is not good for you. Last here, we have the inner ear, which is this whole thing there. And, you know, different parts have different names, which we'll get to in the next diagram. So there you have it. Basic stuff associated with the ear. Okay. Let's move on to this next one. So we are zooming in the middle ear. And again, we will see that there is the tympanic membrane that we labeled previously. Tympanic membrane. Okay. And connected to the tympanic membrane on the medial side, we have one of the three ossicles. So that's the first one. And then we have a second one here. And then we have a third one right here. Okay. So these ossicles, like they have like a common name, and they also have like a proper biology name, if you would. Okay. And so what happens is we'll I'll give you both. So first one, this one is called a malleus. Malleus. But people who discover it, like these anatomists, I guess that's what we call them, they're like, oh, that looks like a hammer. Let's call it a hammer. If you ask me, it looks nothing like a hammer. And then the next one is called an anvil. Oh no, so the proper name first. Okay, incus, I-N-C-U-S, and then people are like, no, it looks like an anvil. For those of you who don't know what an anvil is, let me see if I have a picture of an anvil. There we go. That's an anvil. You know, it's like the thing that the blacksmith hammers on, right? That's the anvil. And then the last one, right, looks like the thing that you put your feet on when you ride a horse or give birth, right? That's called a stapes, a syrup, sorry. Let me go back to this here. So the proper name is stapes. And then the one that's supposed to resemble the actual thing, that's the stirrup. I know this looks like it's pronounced stapes, but it's stapes, okay? I promise you. So that is those are the ossicles, okay? They amplify the sound. Just give me 1 second, okay? I just need I'll be right back. Just 1 minute, okay? All right, sorry about that. And so the malleus is connected to the tympanic membrane, and then the stapes is actually pressing on another structure that you can't really see in this diagram. Okay, I mean you can kind of see it. I will outline it. Okay, and that structure is called the oval window. Oval window. Okay. So if we go down here, we can see it better. Okay. We again have the stapes. Or stirrup, and it was pressing on this membrane. That's the oval window. Okay, what does the oval window do? I will explain in a second. Okay, it's kind of like a membranous structure. Okay. All right, let's go back to here for a second. This tube, you remember this tube? We labeled it previously. That's the Eustachian tube, which is again responsible for equalizing pressure with the surrounding. And then all over here is just like empty space. Okay, so we call that the tympanic cavity. Okay, so that's pretty straightforward so far. I'll tell you what the oval window does in a second. Okay, so label more stuff here. A little bit below, underneath the oval window, is another window. We apparently have a lot of windows in the ear. This one is called the round window. Round window. Okay, and they're related to each other in terms of functions, which again, we'll get to in a second. This snail shell looking thing, it's kind of cool. Every time I see these pictures, like, oh, we actually have a snail shell in our ear. And that is called the cochlea. Over here, all thing. Nice little snail shell right there, a little bit of escargot. That's the cochlea. Okay. Now, the thing you have to know about the cochlea is it's a bony structure and it's filled with fluid. Okay, we're not going to worry what the fluid is called. It's just fluid. All right. And I guess we can color a little bit more of it. Okay. There we go. All right. I'm gonna try to explain this. Right, so remember the oval window? The stapes is gonna press on it. And then we have the cochlea, and then we have the round window. So let's go back to our class note here, which I will post later on. So this is the cochlea. Oh boy, this is the cochlea. All right, and I'm just gonna very roughly put the which window is that? Again, just want you to think about it. That's the oval window. And then we have the O here, which is the round window. Okay, and then we have the stape here pressing on it. That's the stape. It's gonna be pressing on it. Now it's really hard to draw it, you know, in a coil form. So one way to visualize this is think of like cinnamon roll. I don't know if you guys ever made a cinnamon roll, or you probably have eaten one, right? Cinnamon roll. It actually is a long roll of dough, right? Like this. Okay, and then you spread your cinnamon on there, and then you roll it up, right? You roll this up, okay? And then you get the cinnamon roll, right? Can you guys visualize what I'm saying here? Okay. So this is like that, the cochlea. Right? We are gonna unroll the cochlea, right? And it's gonna look like this. Okay, gonna unroll the cochlea. It's gonna turn out to be this very long thing that tapers off. Okay, so if you imagine taking this and then you roll it up, roll it up, roll it up, roll it up, and then you will get back this spiral. Okay. Now the inside of the cochlea is actually separated into three channels, okay, three tunnels, if you will. Okay, so there is one tunnel at the top, and then there's one tunnel at the bottom, and then there's one in the middle. Okay, like that. And they all have names, so we're not going to worry about it. Okay? So this is going to be the beginning. That's where the oval window is going to be. And you're going to have your stape. You're going to have your stape pressing on it. Okay. Why is it pressing on it? Well, because as the sound strikes your tympanic membrane moves, right? And then that causes the hammer to transfer the movement to the anvil, which then transfer the movement to the stapes, which then will be pressing on the oval window. Now, this whole thing is actually like the channel actually extends inside, right? Okay, like this, and it's filled with fluid, middle channel as well, but I'm not going to draw it because it will make the diagram very confusing. And then we also have a bottom channel here, like that. And this whole thing is filled with fluid. Okay, so at the bottom here, we're going to have the round window one more time, round window. Okay, so as the ossicles are moving and the stape presses on the oval window translate the movement of the stapes into pressure waves in the fluid. So that's going to create a wave that goes forward. Let me just use a different color. Sure, this one. Okay, wave's going to go forward. And depending on the pitch of the sound, like high pitch or low pitch, depending on the pitch of the sound, is going to turn around at different places. And then the round window is going to bulge out. So when you press in on the oval window, the round window bulge out to relieve the pressure that's building up in the cochlea. Right? You understand that? So like this hand is oval window, press in, and then the round window bulges out. And when the oval window goes back to normal position, the round window will go back in. So they kind of go in like opposite direction to accommodate for the pressure that's building up inside your cochlea. So the function of the round window is to relieve excess pressure in the cochlea. And the function of the oval window is to translate movements from staypiece into pressure wave that goes into the cochlea. Does that make any sense? Let me know if you have questions, please. All right. So the middle layer, which I haven't been drawing much to it, right? That's where the hair cells are gonna be. Right? So there's the hair cell, hair cells, hair cells, hair cells all the way. Okay, more hair cells here, hair cells, hair cells. Okay, so the hair cells, the mechanoreceptors, are located in a structure called the organ of Corti. Organ of Corti. Right? So depending when the wave turns around, hair cells of that particular location will be stimulated, and then it will send a signal to your temporal lobe. Remember, temporal lobe is where you process auditory information. The best way to imagine this is think of like a piano keyboard. Okay? So when the sound turns around here, it presses this key, ding, and then it creates a particular sound. Right? If you have a sound of a different pitch, it will go maybe deeper and then turn around later, and then you will stimulate those hair cells, and then your brain would interpret it as a different pitch. Okay, so lower frequency tends to travel deeper. Lower frequency. Okay, lower frequency is lower pitch. We'll say lower pitch. Lower pitch. Over here, that's like higher pitch. Higher pitch. Okay, higher pitch like wee, like those sound, right? Lower pitch like okay. So lower pitch goes further down, higher pitch is closer to the outside. As we age, we actually lose the ability to detect higher pitch because the hair cells that are closer to the outside of the ear, actually they get damaged. Especially if you like, use your earbuds a lot, right? Play them really loud, right? Then that's gonna damage the hair cells closer to the front, to the outer portion of the ear. And when you get older, you won't be able to hear higher pitch sound. Okay. So that basically is what the ear does in terms of detecting sound. Okay, you can read the PowerPoint notes later on your own, but I think I summarized everything that you needed to know. Last call. Any question before we go back to learn more? Okay. So the other function related to the ear is detecting motion of the body, which in turn help maintain balance. Okay, so there are two additional parts in the inner ear that we need to know. So this thing in the middle, this middle chunk here, that's the vestibule, which is responsible for detecting acceleration Okay, so there are two types of acceleration. Horizontal acceleration, which is like, imagine in a car, when you're speeding up or pressing on the brake, right? That's horizontal acceleration. And that is detected by this part right here, this lower part. Right, this lower part right here. That's called the saccule. Right, this is for horizontal acceleration. Okay, again, like in a car, e.g. in a car. Not when you're just traveling at like, 60 kilometers per hour, then the saccule doesn't really get stimulated because you're not changing your speed, right? Okay, it's only when you are speeding up or slowing down. Okay, then your saccule will detect that. And then the other kind of acceleration is vertical acceleration. That's like when you are dropping, when you're falling, and that's detected by this thing right here, another part of the vestibule. That is the utricle. Right, so this is for vertical acceleration. Example, falling. Okay, so in general, the vestibule is responsible for detecting acceleration. If you think about on a roller coaster, then these two things will constantly be sending signals to your brain, right? Because you're both accelerating vertically and horizontally. Okay, someone asked me what is motion sickness. Okay, interesting. Motion sickness is when the information that is being sent to your brain does not agree. Sorry, let me rephrase that. Motion sickness occurs when the information that your brain receives from the inner ear does not agree with the information it's receiving from the eyes. Okay, and that creates motion sickness. Example, if you're reading in the car or reading your phone, looking at your phone in the car, you might get motion sickness because what you're reading, your eyes is reading on the phone, and those things are not moving. So your brain is receiving information from your eyes that you are not supposed to be moving, but your inner ear is sending all these signals to the brain, telling you that, oh, we are accelerating, we're stopping, we're going forward, backwards, right? And the brain get confused, and that creates the sense of motion sickness. Same thing for playing video games, right? For some people, right? Your character is moving in the screen, but then you physically is not moving, right? So it's like the other way around compared to in the car. The inner ear is sending signal to the brain that tells you you're not actually moving, but your eyes is sending information to the brain and tells you that we're in motion, right? And that creates the motion sickness sense. But some people don't get motion sickness. Some people are like immune. Some people are very prone to it. I remember like on our honeymoon, we went to this like excursion where we have to take a boat ride to some ruin, like some Mayan ruin, I think. Okay, and we were so sick by the boat ride from the boat ride. By the time we get there, we literally did nothing, right? So we just wasted all this money just to suffer. We joke about it now, okay? But on a boat again, like look, those walking back and forth, right? Okay, those are the signals from the inner ear. Your eyes looking at stationary stuff inside the boat, right? Or even if you try to look far away, because nothing is moving. If you look out to the water, right, and the sky, everything looks stationary, and you get motion sickness. Okay. The other thing is this stuff right here. Okay, that stuff is these are called what is it called? These are called semicircular canals. Semicircular canals. And I need to show you in the 3D models to help you understand it a little bit better. All right, so here we go. All right, so this is the inner ear, and let me see if I can isolate this thing. Isolate, there we go. Okay, so this is in its normal position. By the way, like if you wonder where this thing is, like in your head, like if you have a hard time visualizing where it is, like it's right here. Like it's just underneath your eye. Okay, if you go all the way in, like maybe about 1/3 of the way in, you're gonna hit this thing. Okay, so that's where it is. So we have the semicircular canal, and there are 3 semicircular structure associated with it. Okay, so again, think of the room that you're in right now, assuming you're in a room. The wall that's in front of you is going to be parallel to this canal. Okay, that's called a posterior canal. This one here at the top, that's going to be parallel to the wall on your sides. Okay, and then this one here, that's going to be parallel to the ceiling or to the floor. Right, so the 3 of them together kind of forms like a perpendicular to each other, right? So like if you think about, again, like a corner of your room, right? Corner of the room, like this is the corner of the room. You will have one canal that's parallel to this wall. You will have one here to the floor and the ceiling, and then you will have one here. Does that make sense? So what do they do? Well, when you, oops, how do I go back? Clear. Oh, I don't know how to go back to here. Oh, there we go. Full screen. So these semicircular canal will detect head movement. When you nod your head, say yes, like that, right, up and down, then this canal will be stimulated and it will send signal to the brain. When you shake your head, say no, like that, no more anatomy, then this one is going to get stimulated and it's going to send signal to the brain. What about the one at the back? What kind of motion do you have to do to stimulate that one? This one at the back. How do you move your head to stimulate that one? Okay. Like tilting your head to the side, right? Like that. I can't do it on this model here, but if you tilt it to the side like that, then the back one is going to get stimulated and it will send signal to the brain. So basically, these are the 3 canals. Let's color them. Okay, so we start with this one. This one is the anterior one. Anterior semicircular canal. Okay, I'm just going to use short form here. And to stimulate that one, you know, it's like nodding your head as in saying yes. And then we have the posterior one. Semicircular canal. That's like tilting your head to the side. Okay. Just like tilt head sideways. Oh, you can't see that, can you? Tilt head sideways. And there's one last one, which is the that is the lateral semicircular canal. And that is like saying no, shaking your head. All right. That is it. Let me go back to this diagram here. I'm going to add one more thing to it. There's going to be a big nerve over here that takes all the information to the brain. And it takes information not only from the cochlea, but also from all these other stuff that detects motion of the body. So it's called the vestibulocochlear nerve. Okay. So information from the cochlea, because it's related to sound, it would get sent to the temporal lobe. Information related to body movement, acceleration, right? That's gonna get sent to the cerebellum, okay, for balance, right, and equilibrium. Okay, so all the stuff in the PowerPoint you can just read on your own. It's basically what I told you, and you know, just read through the information. Just a couple more slides here, though. There is something called synesthesia, which is a condition where the ascending path that goes up to the brain from different senses gets jumbled up. Okay, you're born with this. It's not something that you develop in your life. Okay, and so for these people, even though the colors, like even though the words on a page is black and white, they will see color to it. Okay, and that's the color that they will see. Okay, so there is a page that's kind of cool. I want to show you. You can type your name and see what color it is. So if I type my name, okay. So for someone with the synesthesia, when they read my word, the V will look like this color and the I will look like this color, and it would just have like a particular color code to the name. Okay, you can try your own name to see what it would look like. And you know, in this particular picture, I want you to see if you can find out the twos. Okay, I mean, if you look at it for long enough, you probably will be able to identify the twos, right? But for someone with the condition, they will be able to see it right away, because for them 5 is blue and 2 is red, right? So in something ordinary to people who don't have the condition, it's hard to pick out, but for them, they will be able to see it right away. It's kind of cool, right? But it's not just for letters and colors. Depending on what form you have, some forms will mix taste and letters together. I read about it. Like so when they see certain words or certain letters, they perceive it as a particular taste. Okay, so I'm just making this up. When they see the letter F, they're like, oh, that tastes like broccoli or something. Okay, and sometimes it could be certain shape associated with a particular scent, right? It depends on which path gets mixed up, right? And it's really interesting. You probably should look it up and read about it. Okay, we just have a few questions here. I will just go through it with you. Which of the following presses on the oval window? If you have been paying attention, you will know that is the stapes. Stapes. You are shaking your head side to side as in saying no. Which semicircular canal is stimulated? Okay, side to side. Just look at the notes. You will know it's a lateral one. Another question I like to ask on tests is this, okay? Like, instead of like saying yes, saying no, in terms of head movement, sometimes I ask you, like, what if you were doing a cartwheel? Okay, do you guys know cartwheel? Cartwheel like this. Okay, so like that. I'm trying my best here. Give me a second. Okay, and then you get it, right? That's the cartwheel. When you're doing the cartwheel, which canal is stimulated? Okay, you should think about that. Okay. So doing the cartwheel is rotating this way, and the answer would be the posterior semicircular canal, right? Because this is kind of like tilting the head to the side, right? That's the posterior semicircular canal. What about lateral? What kind of motion do you have to do to stimulate the lateral semicircular canal? Give me an example. This is called cartwheel, by the way. Cartwheel. For people who don't know. What do you need to do to stimulate the lateral semicircular canal? Looking back. Oh yeah, like that. Okay, yeah, sure. But like in terms of like something that's analogous to the cartwheel, eh? You see this in the figure skating a lot, or like ballet, right? Like if you were to spin like this, right? Okay, like spin like this. I can't do it. I'm gonna get all tangled up in my quads. Okay, but that would be like a pirouette. Right? That's what it's called. Right? So that would stimulate the lateral canal. And as for the anterior semicircular canal, it would be like doing a somersault, right? So like rotating this way. All right, something to think about. Right? If you're confused, just let me know. We can explain it again. You are sitting in a car as a passenger. The driver slams on the brakes suddenly to avoid running over a squirrel. Which structure in the year detects this motion? So this is considered as linear acceleration. Okay? Some people think acceleration is always speeding up, but slowing down is also acceleration. And we would need to have the vestibule for that. Okay. So when this fluid in your inner ear gets messed up, or like it's not functioning correctly, I should say, to put it in more proper terms, then people can get vertigo, right? Yeah, exactly. And Min beat me to it in the chat box. So like these people would be like sleeping on the bed, and when they open their eyes or something, like they feel like the whole room is spinning, right? So like these structures, the semicircular canals and the vestibule is sending signal to the brain, even though you're not stimulating them. Okay, and that will cause you to feel very, 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 very ill. Okay. Anyway, we end up going all the way to 11. I'm sorry. We will continue on this next class. There is a very important diagram like this one that we are gonna fill in together. I mean, I will post the answer key as well. But I think we will do it together as a review next class before we start on the next lecture. Just before you go though, let me just quickly check to see when the test is going to be. Because honestly, I totally don't remember. Let's see what's going on here. Oh, this is the wrong class. Sorry, just stay put for a second. Okay, so we are here right now, Wednesday. Okay, so next week we will do digestive system. We honestly, we probably won't be able to finish the whole thing by the Wednesday. I think we might have to move the test one class later. Okay. I'll make an announcement for that. Okay, but we will do digestive system on Monday. We probably will be able to finish up digestive system and take up the lab. But we might not be able to do a review. Okay, so let's just make it official. Okay, I'm gonna move the test to the 22nd instead. Okay, the test to 22nd. If that's okay with you, I'm sure it's okay. All right, I'll post the announcement to make it official. All right, have a good week. Thanks for coming. Bye.